Okay, these are your notes over mutations and denaturing. Essential question, how is protein function affected by mutations and denaturing? This is our outline. First we're going to talk about denaturing, then mutations, the different causes, effects, and types of mutations. Okay, first, blank. Proteins have a very specific shape. This right here is your line of amino acids, and then it alpha helixes or beta pleats, and that's your secondary structure, primary structure, secondary, and then a bunch of those secondaries combine to make a tertiary, and then that right there goes to that right there, and then several of those combine to make your quaternary structure, and then the substrate that's going in there has to fit very specifically right in there. So if you mess up that shape, the protein is not allowed to do its job. Okay, certain things in a cell and your body affect proteins, like temperature, salinity, and pH. If these are not in the right range, the protein will denature. Change in a protein's shape or nature so that it cannot function. So you've got it all wound up, the protein right here, and then if it's too hot, it's going to denature and just go to its primary structure as a line of amino acids. Okay, linked to previous topic, this is why homeostasis is so important, so the proteins can do their jobs. If you are outside of homeostasis, then your protein will denature and you're not able to do your job. Okay, that was denaturing. That was when you have a protein and then it just is not in the right shape. Mutations are a change in the DNA which changes the protein. Okay, so a mutation is any change in DNA. There are two different causes, a mutagen or a spontaneous cause. A mutagen is anything that directly changes the DNA. So UV light, like this tanning bed, chemicals, radiation, and that is the cause of the mutation. A spontaneous mutation is a random change in DNA. There was no reason or cause. It just randomly happened. So it could be a random mistake in the copying of DNA. Like how a copy machine sometimes randomly messes up when it's making a copy, and then that mutation is going to be seen in the protein when you make the protein. Okay, there are three effects of a protein. Bad, good, and neither. A bad mutation will cause disease, cancer, or deformity. A good mutation would increase the chance of survival of that organism. So if you look really closely, can you see it right here? Can you see it? And then, can you see that? And that? Okay, so those are mutations that cause different colors of those three moths right there. We'll study that more when we get to the evolution unit. Okay, if the mutation increased the chance of survival, then that is good. If it made it faster, stronger, smarter, whatever better adapted means for that particular organism in that particular environment. A mutation that is neither good or bad, the mutation brought in the same amino acid, so there's no change in the protein. So when we did our protein synthesis, if it mutated from GCG to GCA, it brings in the same amino acid, so there's no change whatsoever. It's still a mutation, but there was no effect. And that is called a silent mutation. Cross out extra if you're in an honors class, because this is not extra. You got to know this. You can do it though. There are three types of mutations: a deletion, insertion, and substitution. In a deletion, a nucleotide, the A, T, C, or G, was lost or deleted. So right here we took that out. An insertion is adding a new one. You can kind of see that little C that we added in. And then a substitution, think a substitute teacher. So we didn't add or delete a teacher. The teacher is still there, it was just substituted with a different teacher. So one nucleotide is substituted or replaced by a wrong one. Okay, next slide we're going to talk about how bad these mutations are, and focus on how the red mRNA corresponds with the red amino acid there. Okay, let's start from the bottom. Again, if you're in honors, cross out that extra. So starting at the bottom, if you substitute one letter for another letter and nothing happened, that is called a silent mutation. And that is neither good nor bad because nothing happened. If you missed the amino acid, you missed the right amino acid, that is bad because that brought in a different amino acid, and that's going to change the protein. But it's not that bad because it just affected one, and all the rest of them will still be the exact same. If the substitution codes for a stop codon, that is going to be very bad because now the whole rest of that protein is no longer coded for. Okay? A deletion or insertion is going to shift your reading frame. So we go 3, 3, and it's going to shift that frame one way or another. So every single amino acid afterwards will be changed. And that is very bad because then pretty much your whole protein after that is going to be different. Okay. You now have Quizlets and a worksheet practicing all these things that we just learned.